Hello and welcome back to my channel. So in this part of the video we are going to discuss a new series, that is translation in prokaryote. So in the previous thing we have discussed what is replication and from how replication the DNA is replicated. Then we have discussed about the transcription, how from DNA, mRNA or RNA are formed. We have both discussed in case of prokaryote as well as eukaryote, and some post transcriptional method also we have discussed. Now we are going to discuss about the protein synthesis, that is the translation process. So what is translation? Translation is the formation of protein from the RNA. Okay, and you know very well that where does the translation process takes place, that is in the ribosome. You know that prokaryotes has 70s type of ribosome and eukaryotes have 80s of ribosome. I have already discussed about them when we were studying about the structure of RNA. If you have not understood what is the structure of RNA, what are the different types of RNA, and what are the role of these three types of RNA, first I refer to you to please watch that video. Then you can watch the transcription series, then you can see this video for the better clear understanding, because this is a central dogma, or this is a clear process you can say. Now you must be wondering what is central dogma. Okay, central dogma means that is the DNA to mRNA, that is known as transcription, and mRNA to protein is known as translation. This is known as the central dogma. Okay, the central part of the molecular biology, that is every protein is synthesized with the help of the DNA as a template. Why I am saying so? Because DNA provides a template for the RNA synthesis, and RNA will be formed from the which will form to provide the protein will be formed. So in short we can say that DNA provides a template for the protein synthesis. So this is all about the introduction which we have been discussing till now, but now come to the major topic, that is translation. In this first part we will be discussing about translation in prokaryote. In next part we will be discussing about the translation in eukaryotes. So let's begin. Before that, you must know that is major difference between a prokaryote and a eukaryotic cell. You might be knowing that the prokaryote lack nucleus. Okay, so that's why you can see here this is the prokaryotic cell and this is a eukaryotic cell. Okay, wait 1 minute. This is a prokaryotic cell and this is a eukaryotic cell. You must be seeing in this that the prokaryotic cell lacks nucleus, whereas eukaryotic cells have nucleus. So by now you must have been guessed right that in prokaryote, that is the translation and the transcription both takes place simultaneously here only in the cytosol only. Okay, and in case of eukaryotes, the translation, that is the transcription process, takes place in the nucleus. You can see here after the processing, that is here this is from the DNA. From the DNA the mRNA is formed, which is known as transcription, and then after processing, that is post transcription, that is mRNA, mature mRNA form. Then these mature mRNA from the nucleus is transported into the cytoplasm for the process of translation. Okay, we will be discussing this eukaryotic part in the next part of the video, but this is the major difference between the prokaryotic translation and the eukaryotic translation. Prokaryotic translation takes place and transcription takes place simultaneously, whereas translation in case of eukaryotes, that is transcription takes place in eunucleus, and then it is transported into cytoplasm, and then the process of translation begins. So this is the difference. Then let's study the process of transcription. Before that, translation. Before that, you need to understand that what are the things take part in the process of translation. As you have discussed in the transcription process, RNA polymerase was required for the transcription, promoter region was required. Then the three processes, that is initiation, elongation and termination, was required in transcription. But what are the requirement for the translation process? First requirement is the mRNA. Obviously mRNA must be there so that the protein synthesis will be formed. Next is ribosome, obviously because a protein synthesis will take place there. Next is tRNA. When we were discussing about the structure of the tRNA, I have already discussed that the tRNA is known as transfer RNA. That is, it transfers the amino acids to the mRNA for the protein synthesis. Okay. Next, obviously, proteins will be formed. Next, what are the three major steps? That is initiation, elongation and termination. And here also the translation always directs in the 5 prime to 3 prime direction. Okay. Next, you can see here, this is the overall process. I am just telling you, this is the small ribosomal subunit and this is the mRNA. So the small ribosomal subunit will bind to the mRNA in the initiation site, and then the tRNA will come and bind to that, and the tRNA anticodon sequence, you must be wondering what is anticodon sequence. In the structure of tRNA, I have already discussed there are 5 arms of tRNA. Among that 5 arm, one arm was the anticodon arm. If you remember that, if you don't remember, do watch that RNA video for the better understanding. So the sequence of the anticodon tRNA is complementary to the sequence of the mRNA. Okay, this is very important for the attachment of tRNA to the mRNA, so that proper binding will take place, and then the protein which tRNA comes will be binded properly. So this is the process. And next, what happens, the tRNA which will carry the protein, that is the amino acid, will come and bind it, and the protein synthesis will begin. We will be studying each one of them in detail. Let's begin. First is the initiation step. I have made a note regarding this translation, so I have put this picture here only, so that you must be clear with that, because in book it was really confusing, little bit confusing, so properly I have made a note for your better understanding. So what happens, this is the initiation step. I forgot to mention, but you can refer, this is the initiation step. Okay. So the initiation steps, what are the initiation, what happens in initiation, let's study each one in detail. So as you know that the prokaryotes have the 70S or 70S ribosome, okay, and you know that the 70S ribosome has two subunit. One is the 30S, another one is the 50S, and 30S is known as the smaller subunit and 50S is known as the larger subunit. Okay, you know that properly. So this is the structure of the ribosome. This is the 30S, that is the smaller subunit, and this is the 50S. In this larger subunit as well as in the smaller subunit, there are three type of space is present, which is known as E site, P site and A site for the binding of the tRNA. Okay, what is this role, we will be discussing later on. So E site, P site and A site will be present in the ribosome. 70S ribosome RNA has two subunit, as I have discussed now, that is larger one is 50S and smaller one is 30S. Okay. Next, what happened, initiation factor, that is IF3, binds to the E site so that the large complex of the ribosome does not bind to it. Now you must be remember that first what happens, these two subunits need to be separated out, because when will this larger subunit come and bind, when there will be proper initiation complex will be formed. After that the larger complex will come and bind, and then the process of elongation will begin. Before that only the 30th subunit will take the role in the initiation, and here three subunits are three structures are present, that is known as E site. Three sites are present, that is known as the E site. Wait 1 minute. This is the E site, P site and A site. What happens? Certain factor, that is IF3. What is IF3? IF3 is a initiation factor. So IF3 initiation factor will come and bind to this E site. Why? So that it will block the E site, so that this large subunit, that is the 50th subunit, doesn't come and bind here. Okay, it prevents from binding to this region. Next what happens? Next what will happen? IF2 Remember this sequence. Okay, next what will happen? IF2 will bind to the A site so that the tRNA which brings the first amino acid binds to the P site and GTP is used. Very, very important point. What happens? First the IF3 will block this E site. Okay. After blocking the E site, next factor that will come is the IF2. Okay, this IF2 will come and block the A site so that the tRNA which will come will bind to this P site only. It will not bind to the A site, it will come and bind to the P site, and here the energy is used, that is the GTP. So remember this point, that the first amino acid which will come will block the P site only. Next, the amino acid which will be coming will be occupying the A site. Okay, this must be remembered. So the first amino acid which will come will occupy the P site. Next, what happens? The IF2 brings the charged tRNA, that is the first amino acid, that is formylmethionine. Formylmethionine is the first amino acid in case of prokaryote. Only methionine is the amino acid in case of the eukaryote. Remember that. First amino acid is formylmethionine. Okay, F-methionine tRNA, and binds to the P site and forms 30S initiation complex along with that of the mRNA. So what will happen? Here this E site was occupied by IF3, A site was occupied by IF1. Now what will happen? This IF2 will bring the tRNA and they will bind in this P site. How it will bind? I have shown you in this picture. That is, this anticodon of this tRNA will form a proper binding with this mRNA structure. So now the initiation proper mRNA will come and this tRNA will be there. Now this is the proper initiation complex will be, that is the 30S initiation complex is formed. Okay. Now what will happen after this initiation complex is formed? Now the largest subunit will come and bind to the 30S region. After this one, the IF2 and the IF1 will be released, because there is no need of this IF2 and IF1. IF2 was only needed for the binding to that of the binding of the tRNA to the P site, and IF1 was binding to the A site. After the T site has been occupied by the P site, the IF1 and IF2 will be released, and here the GTP is converted to GTP. Now the initiation complex, that is total 70S complex is formed. Okay. Now 70S initiation complex is formed. Okay. Now this is the end of the, that is the initiation process. Okay. Now the initiation process is completed. Moving to the elongation, what will happen? First amino acid was occupied by the P site. Okay. Next what will happen? In the step 2, the second amino acid which comes bind to the A site. The binding of the tRNA to the amino acid is done by the enzyme, that is amino acyl tRNA synthetase enzyme, and along with that certain other factor, that is EFTU. EF means elongation factor TU. GTP helps in binding of second amino acid to the A site. So P site is already occupied, as you have seen. You can, this is the mRNA, this is the 30S subunit, this is the 70S subunit, this is the E site, this is the P site which is already occupied by the first. A site is now empty. Now the second amino acid which will come will be binding to this A site. So the binding is mainly helped by the one type of enzyme, that is amino acyl tRNA synthetase enzyme. Along with that certain elongation factors also help, that is EFTU. Okay, which use GTP as an energy source for the binding of the second amino acid. Here threonine is bind. Okay, first here was methionine was bind, now here threonine is bind in the second side. Now what will happen? After binding, the GTP will be converted into GDP. Okay, next step 3, what will happen? This A site transfers amino acid to the P site due to the movement of ribosome by one codon, and the position of each gets shifted and the A site becomes vacant. So what happens? After the binding, wait 1 minute. Okay. So what happened? After this A site binding of the second amino acid, this amino acid will be shifted to the P side. Why this happens? Because there is certain another type of the enzyme that is known as the or another type of protein which is responsible, that is EFG GTP. EFGTP G is the elongation factor G. What will happen? It will shift the ribosome by one codon. It will just move the ribosome. As a result of moving, this amino acid that was occupied by A side will be now shifted to the P side, and then what will happen? This amino acid which was present in the P side will be occupied in the E side. You can see here. Okay, what was present in the P side will be moved to the E side, and whatever which was present on the A side will be moved into the P side. This movement is done by the EFG GTP. Hope you are clear with it. Next, what happens? After EFTU GTP transfer, the charged tRNA to the A side, it is converted to EFTU GTP. To convert it back into GTP, EFTS protein is required. So, you can totally say that there are three proteins which are majorly take part in the elongation process. First is the remember this, EFTU. What is the function of each? I am going to discuss now. EFTU uses a GTP. What it will do? It will bind the second amino acid. Okay, it will bind the second amino acid to the P side. First one this is. Second one is the EFTG. Okay, EFTG. Oh, sorry, EFG. Okay, what is this role? Its role is to move the ribosome by one codon so that the positions of each of them get shifted. Then third is the EFT. Okay, next third is the EFTS. Okay, EFTS, what is its role? After the binding of the tRNA, okay, after the binding of the tRNA, it was converted back to GTP. To convert back to GTP to the GDP form, this EFTS is required, because another amino acid will come, another GTP will be used to move back to the GTP form. This EFTS factor is required. So these are the very, very important three elongation factor which plays an important role in the process of translation. Now the process of translation is elongation is completed. As the ribosome keeps on moving, when it will come to a sequence called UAA, UAG, or you can say UGA. These are the three termination codon. Okay, these are the three termination codon. Then a releasing factor will, that is factor 1 and factor 2 will be released, and it can no more elongation take place. And RF3 is the, what is RF3 is the that is releasing factor 3 which uses GTP, and it will break the bond of the, that is amino acid with that of the tRNA, and the protein will be released out. So this is how the translation of the protein takes place in the prokaryotes. Next, total, if you understand how many GTP are used, it is asked sometime in exam. One is the one GTP was used during the initiation by the IF2 when it was binding to the E side. Sorry, when it was binding to the A side. Okay. P side, when it was binding to the P side, one GTP was used. Next, 2 GTP was used during the elongation process. You can see that is the EFTG and EFTU. This both have used GTP, 2 GTP. Okay. Next, 1 GTP was used during the termination, that is by the RF3 factor. So total 4 GTP is used. Okay. Next is RF1 and RF3 are the other components is also released. So these are certain releasing factor. This is all about the translation process in prokaryote. Hope you are clear with this. If you have any doubt, you can ask in the comment section. Do you share among your friends? This is a very important chapter, and very usually they have confusion. Students have confusion regarding this topic, what is this process, and they find it complicated, but I have tried to make it simplified version. If you have any doubt, you can ask in the comment section, and share as much as you can. And if you have not subscribed to my channel, do subscribe, and like if you find it informative, and also do watch the previous video for the better understanding. If you directly see this video, if anybody have no idea regarding anything about molecular biology, they might find it very difficult. But if you are new to this field, if you don't know what is molecular biology and all, I request you to first watch this from the starting video, that is DNA structure, then all, then come to this part. If you also have any idea regarding what is DNA RNA, I still request you to watch from the beginning, because there are certain important things I have discussed regarding exam point of view, which is very informative. And thank you. That's all for today. In the next part of the video, we will be discussing about the translation in eukaryotes. Till now, have a nice day. Thank you.